The private chat first. We are in private chat.
So it's live.
Yeah, okay.
Everyone can see you and hear you.
So good afternoon, everybody, for this webinar organized by Society of Radiation Research. The webinar is on genetics in cancer and its therapy. We have our guest with us on the platform here. Let me introduce our guests.
इधर से देखिए.
We have Dr. Vijay Singh with us, Dr. KP Mishra, Dr. B.N. Pandey, Dr. Shyam Srivastava, and Dr. Rajiv Sarin. So let me first call upon Dr. Shyam Srivastava to give his welcome address. Dr. Shyam Kishore Srivastava is President SRR and ex-head Department of Radiation Oncology, Tata Memorial Hospital, Mumbai, and Director Radiation Oncology, Apollo Hospitals, Navi Mumbai. Sir, please deliver your welcome address.
Yeah, I think I'm audible. Thank you, Pooja. And good afternoon, everybody. At some places, it may be good morning. And I would like to welcome everyone. And we all know that in this difficult time, there are several problems that we all have been facing. It was very difficult for all of us to meet each other. And I think this was the one wonderful media where we all can really see each other and exchange. I remember when the lockdowns were there in various countries, we were not able to meet each other and the webinar started. And I remember in the very beginning when the webinars were there were large number of people who were joining. But as the number of webinars kept on increasing over last 4 to 5 months, the number of people who were attending was much less. And as I said that initially maybe 1,000 people were joining, and I have attended few webinars just last week or something where hardly 25 people were there. But I am very happy with the Society of Radiation Research that a lot of work is being done, and it has been done in last few years. And I am thankful to Dr. Badri Pandey, who are really very actively working. And I am also thanking to my previous president of the Society of Radiation Research, Dr. KP Mishra and Dr. Nagaraj Hulgul. Because of them, this has been kept very alive. And I am very happy today to really welcome you all for this webinar, which really has been attended by a large number of people, which is quite unusual. And I must thank Dr. Sarin, who is my colleague, that he has put a subject which is really is being interest to several people. So I welcome you all, and I'm thankful to Dr. Vijay Singh, who is joined us from the Maryland in USA, and he will be introducing Dr. Sarin. So once again, welcome to all of you, and I'm sure this webinar will be extremely useful to all of us. Thank you.
Thank you so much, sir. I would now like to call Dr. KP Mishra, founder, president, SRR, ex-head RBHSD BRC, Mumbai, and ex-VC Nehru Gram Bharati University, Allahabad, to introduce us about SRR and its activities.
Yeah. So, my greetings to all the participants of this webinar, and I'm extremely happy that I'm able to join you this meeting. All those who have been contributing to the activities of Society of Radiation Research, we are very grateful to them. Society of Radiation Research was formed in 2014 with an idea to promote radiation research in the country and also to establish collaboration with international radiation research bodies. And I'm happy to tell that we are now affiliated to International Association of Radiation Research, which is international body, and we are now in lead position in Asian Association of Radiation Research. So, Society of Radiation Research, the way it was conceived, it has delivered, and I'm very happy that a good response is coming from all quarters of the country. And today we have close to 100 membership drawn from various parts of the country, all important institutions. We have the membership, and this membership is constantly expanding. This is really a very happy note. And I'm happy to share that we had planned that we will have international conferences, we will have society meetings, annual meetings, we will also have seminars, and we will have discussion groups, and so on. It's good luck, and I must appreciate my colleagues from VRC, Dr. Hilgol and Dr. Srivastav, and many others, and Dr. Sharma, who are actively pursuing the objectives of the society. Today, we can say that we are growing and we are becoming stronger each day. This is a very happy situation. Uh, I must say we have already achieved two international conferences. We had international school organized wherein we had inputs for practical exposure to students and participants. It was very much in demand. We had to close the entry to people. It was so much attended. Likewise, international conferences that we have held had a very international flavor, and speakers from India and abroad, they all joined up, and it was extremely productive and active meeting. I'm glad to share that we have been having these international schools, which is spread to about 2 weeks, and the schools that we had in the past have been very successful. And we had Dr. Eliakis from Germany who gave the keynote lecture, and now another international school has been planned for September. All of you are requested and welcome to join that international school, which is on DNA damage and DNA repair in relation to disease. An extremely good program Dr. Pandey has prepared at the ISA. I must say that the activities of the society are on move, and we are doing very well. All of you are welcome to join us and keep contributing the way it is being contributed. I'm happy to share that we had started Indian Journal of Radiation Research, which we have renamed and we have redesigned and we have reprogrammed in the name Journal of Radiation Research and Cancer Research, JRCR. JRCR is being regularly published every 3 months, and we have recognition many groups which are citable. We are very close to getting approved in Scopus. We are trying that this journal should go to PubMed soon, and for that I would like to request each of you to please contribute original articles, because we have met many conditions of the PubMed, but one of the conditions is that we should have more original articles published in the journal. So it's my very special request to each one of you to please contribute your original papers so that we succeed in getting it done in PubMed. The international school has been organized for September, and Dr. Pandey has already sent the program, and it is also available on our website. You are all most welcome to join us that time. This will be spread in 2 weeks, and very elaborate program has been made. I must say it will benefit to all young senior people and experts. Our endeavor is basically to provide a forum where young researchers as well as those who are specialists and senior researchers, they come together and they share ideas and discuss, and also provide encouragement to young researchers to come to the field. In this, it is a very successful experiment. It's my strong wish that society should keep growing the way it is growing, and it's my desire that the journal which we are publishing, JRCR. Please support in all possible ways. We have been having many financial problems to publish it, but we are managing somehow. And I must give credit to all my colleagues, Dr. Srivastava, Dr. Hilgol, Dr. Pandey, and also Dr. Amit Kumar, Dr. Sharma, many of you, many of you. I'm sorry I cannot take the names of all, but everybody is so active and cooperative. And I'm proud to see that this is one of the very smooth national society. The way it is working just like a family, the way it is working just like a real, genuine group of experts is very happy note to see. So I personally, as founder president, take pride in presenting to you the brief activity of the society. And I request you to contact our website where a lot of details have been given. And in this webinar, I do not want to stand between you and Dr. Sarin, who is going to give very interesting lecture on genetics of cancer and its therapy. So I would like to again thank you all participants, welcome you to this webinar and request for your continued support in time to come. We have very elaborate program to run out for all the year, and next year we are going to hold international conference, that is 5th Asian Congress of Radiation Research. India is the president of this association, which was founded in 2005, and it had conferences in Hiroshima in Japan, and then in Seoul in South Korea, and in Beijing in China, and now India has been given the responsibility. And a date for 16 to 19 November 2021 has been given, and we welcome you all in this conference. And again, welcome you and my greetings to all of you. Thank you very much. Thank you.
Thank you so much, sir. I will now like to call Dr. Professor Vijay K. Singh from Armed Force Radiobiology Research Institute, Uniform Science University of Health Sciences, USA, to chair this session. Please, sir, proceed.
Thanks, Pooja. I'm delighted to introduce Professor Rajiv Sarin, a very accomplished professional. Professor Sarin is a MD, a first year clinical oncology. He is the former director of ACTREC, which is part of Tata Memorial Center. He is a clinician. He is a scientist. He was trained at Tata Memorial Hospital as well as Royal Marsden Hospital in London. He is currently in charge of Cancer Genetics Unit and Professor of Radiation Oncology at Tata Memorial Center. His research areas are cancer genetics, genomics, and breast cancer management. He is the convener of Breast Cancer Disease Management Group at Tata Memorial Center. He chaired the ICMR committee, which formulated the national guidelines of breast cancer management. He chairs National Apex Committee for Stem Cell Research and Therapy, DST SERB Biomedical and Health Sciences Committee, and also the DBT Chronic Disease Biology Task Force. He has 2 decades of experience in genetic counseling, genetic testing and risk management of adult and childhood hereditary cancers, and somatic genetic analysis of solid tumors. He established the first comprehensive clinical and laboratory cancer genetics program in India, and now that program has more than 9,000 families with hereditary cancer. His group has identified several recurrent founder germline mutations in BRCA1, BRCA2, and DNA mismatch repair genes, and established detailed genotype-phenotype correlation in several hereditary cancer syndromes. His lab has expertise in comprehensive characterization of variants of uncertain clinical significance, including functional genomic analysis. He is the Indian co-PI of International Cancer Genome Consortium. Lastly, I know Professor Sarin very well professionally as well as personally. In 2010, when he was the director of ACTREC, he has extended invitation to visit his institute along with my institute director, Colonel Mark Melanson. And we visited ACTREC and we were there for a few days and we came back with very positive experience. Before I call Professor Sarin, I would like to tell audience that please put your questions under comment and we will take up those questions once Dr. Sarin finishes his talk. Professor Sarin, please.
Okay. So thank you very much. And I will now, just one second. I have to, is my screen visible?
Yes.
Yeah, okay. So good afternoon, everyone. And I would first thank the Society for Radiation Research, Dr. Pandey, Dr. Mishra, Dr. Srivastav, Dr. Huilgol, and Dr. Vijay Singh for giving such an introduction. And whenever I get a chance to speak on genetics and cancer, I rarely say no because this is something very close to my heart. And why? Because although a lot of people do research on genes and proteins and genetics in the lab, this field unfortunately has not made that much of impact in the clinics as it should have been. Hereditary cancers more or less remain a neglected condition, although it is not as rare as I will show later. So today I will touch upon what is genetics, because in this group, I think it's a diverse group. I'm told there are both master's level students to practicing scientists to clinicians. So I think to cater to everyone, so I have kept it a simple talk. Also giving some explanation, because for some things, for what scientists will take it for granted, the clinicians need to understand, and what clinicians will understand very well, the scientists need to understand, and of course the students are also there. So I would explain, and I'm not putting much of our own data in this because it's more of a general talk. So we know the genes, the chromosomes, and their alterations. So gene, basically, it's sometimes, if you ask offhand, what is a gene? I mean, we ask this question in the interview, people struggle to answer. I mean, some people know there is this side, these are the parts of the gene, but essentially it's a functional unit of inheritance. It has a specific structure and sequence of nucleotide in the DNA, and it directs the synthesis of a specific protein, which then has a biological function. And I will talk about some of these things. And any genetic material or things which regulate the expression of these genes may be altered. And when it is altered, not always, sometimes it can have a phenotypic effect. And sometimes the phenotypic effect is so severe that it could cause a disease, or it can become a way of resistance to treatment for that disease. So that is what we will cover in this talk. Genotype, again, this term is used in many sort of context. Essentially, the genetic constitution of an individual. Either you can be talking about one locus, so you're saying the genotype for, say, GST-T1, or collectively, all the loci collectively. So people may have different genotypes. Say Caucasians or Indians, they have a different genotype. And when we talk about the entire genetic constitution, that's what we're talking of genome. So you may have genome-wide changes. So this is it. Then genetic alterations, again, for my clinical colleagues and the students, we all know about chromosomal abnormalities, Down syndrome and so many other chromosomal diseases. Whenever a chromosome is removed or added, invariably there will be a phenotypic effect. It may not necessarily cause a disease, but it may cause a condition or a trait. That is there. When you say pathogenic, means it is quite different. Then abnormalities in a single gene, and I am starting in a different way. So sometimes exons or introns of a gene are deleted, large deletions, or maybe 2 exons are skipped or duplicated. These invariably, because as we said, the protein which is coded by this gene, it will be not normal, it will be aberrant protein. And a protein which is so aberrant will definitely not be able to function normally. And if the gene is involved in an important role, say suppose DNA repair or any other important role or apoptosis or something like that, then because the protein coded by the gene is not functional, there's a high possibility, lifetime risk, that the disease or the pathology would manifest. Then you could have small insertions or deletions, what you call indels. So 3 bases removed or 2 bases removed or 1 base inserted, like del AG or del A, or insertion of C or something like that. So if it is in frame, just 3 bases have gone, then it may or may not have any effect. Just the overall protein would have 1 less amino acid or 1 more amino acid, but may not have. But if it is a frame shift, the reading frame changes, invariably at some place down the line, a stop codon would be introduced, and the protein which will be made, which will be quite small, as you all can see. I've drawn in this diagram. So the protein, if it is altered, a coded protein, then it will not be able to function, and it could be diseased. Then what is most confusing is the SNVs, how to take it seriously or to completely ignore it. A lot of research happens in single nucleotide variation. So most of them are silent, and they are called single nucleotide polymorphisms because they vary. The evolutionary biologists use it sometimes, or in as far as disease or the disease risk modification, very few of these are studied now. And then the missense, that means the amino acid, the coded amino acid is changed because somewhere A has become G. Most of these are also single nucleotide polymorphisms. That means they are different forms of the same, gene, and they don't have any effect. Sometimes they may have a modifier effect, so they may increase the susceptibility slightly to development of disease. Generally, when the gene environment interaction, like metabolizing enzymes and other things. But some of them are pathogenic. That means they can actually cause the coded protein. It could be in the amino acid could be in a region which is like sort of interacting, or DNA binding domain, or some other regions where it is a protein interaction is happening, and it could affect that's this thing. And if it is an oncogene, then it could be a gain of function mutation, so it could be pathogenic. Then splice site, when you have aberrant splicing, invariably the protein would not have normal function, and it will be almost all are pathogenic. And nonsense means introduces a stop codon, almost all are pathogenic. So this terminology, I want to introduce somatic and germline. So some people who are basically working only, they're looking at the genomic DNA which is introduced. So it's a single cellular, in a unicellular, of course, everything is both. What is germline is also a somatic, but in multicellular, organisms, what is there? The germ cells, the gametes, a mutation there is germline. And in rest of the body, if it is there, like in a cancer cell developing in an individual, a mutation in a cancer cell, if it is not in the germline, it is only somatic mutation. So soma means body, and germline, we know what is germline. In terms of genetic variation in disease, so we know about Mendel and Mendelian disorder. Fortunately, these are very rare. Mendelian traits are not so uncommon, but Mendelian disorders or diseases are rare. And the DNA variation is necessary. In absence of that, it will not happen. And it is sufficient to cause considerable phenotypic effect. And I'm giving example of only hereditary cancer conditions like hereditary breast ovarian cancers because of mutation in the BRCA1 or BRCA2 gene. And Lee-Fromeni syndrome, which is because of mutation in germline mutation in the TP53 gene, or the guardian of the genome. And so these are the Mendelian disorder. Then we have the more common ones are chronic complex disorders. So they are caused by the common DNA variation, and any one of these variation in the DNA may not be necessary. Even if it is not there, some person will still develop diabetes or heart attack. Neither it is necessary nor sufficient to produce a phenotypic effect. So each variation has a small phenotypic effect. It's a combination that which causes the effect. So, like suppose say lung cancer or head and neck oral cancers. So someone is a tobacco user and one of the tobacco metabolizing enzyme, the gene which codes for that has some polymorphism or some alteration. It is a null genotype. It could increase the risk. Or ischemic heart disease or diabetes. When we talked about Mendel, we have talked about the pattern of inheritance, and I think this is why I'm addressing to my clinicians, and sometimes even cell biologists, because they're looking at the phenotype of one cell. So dominant and recessive traits and inheritance are as important, and sex linked. And in many human, fortunately in cancer, we don't see sex linked changes. But what we see is a lot of non-Mendelian patterns of inheritance, and they create confusion sometimes because the phenotyping has not been done, and there are many other things like genetic anticipation, incomplete penetrance. There are several reasons why it does not look like a Mendelian pattern of inheritance, autosomal dominant. Most hereditary cancers are, by the way, Mendelian autosomal dominant pattern of inheritance, where only one allele is mutated, and that is sufficient. Whereas in recessive, both the alleles, so it has a homozygous mutation, it is a heterozygous mutation. So phenotype is very important. So far we were talking of genetic alteration. So phenotype is essentially the appearance or what you can observe, the observable nature of an individual. And when I'm talking of individual, people who work in the lab, for them, that individual may be not a human being or it could be a mouse. It could also be a cell. How a cell looks, how the cell grows, how the cell sort of invades. So it's a phenotype. It's a property or the phenotype of that cell. And generally, the phenotype is the visible effect of the interaction of the genotype and the environment. So for a human being, the environment means, what kind of sunlight. And I'll explain these two pictures, these two young men, or the food that we eat. And for a cell, the environment could be what is the oxygen available, what are there in the petri dish, what sort of nutrients you have put or starved them. So there's different things for environment. And based on that, the cells might become elongated, or they might sort of become sort of spindly, or they rounded, or they shrivel, or they die. So many things could happen. So the picture I'm showing here is this man has no pigmentation on his skin and his hair, and eyes I have blocked because it's a public lecture. And this man is actually, you see, hyperpigmented, and his nose has sort of gone off. So both of them have UV or the radiation induced skin cancer. This man has cancer over the earlobe, and this man has cancer over the eyes. I've blocked his eyes. Multiple cancers, skin cancers over the nose, because of which multiple surgeries his nose he has lost. So the effect, so what I wanted to explain, that if you look at the phenotype as cancer, the genotype is completely different for these two individuals. So this individual has has oculocutaneous albinism, or OCA, albino. You must have seen these albinos. They have no pigment anywhere in the skin. Oculo means in the retina, in the iris, or hair, everywhere. And that's because of certain mutations in genes that code for proteins that give us melanin or the pigment, different pigments that we have in the body. Whereas this man, who has dark and dry skin, actually skin is dry, has another rare condition, and both of these are autosomal recessive condition known as xeroderma pigmentosa. A lot of you know Xerox. Xerox means dry, like xerophytes or whatever. So because they have dry skin which is pigmented, and here in this individual the cancer was happening because there was no pigment. The same UV light which is absolutely okay for you and me, more of it goes inside his skin and causes more DNA damage. This gentleman, his body has produced more melanin, try to screen or stop more of UV from going inside, but whatever little UV rays which goes in, the damage caused by the UV, the dimers which are produced, his cells are unable to repair that. So the repair excision pathway. So, when I'll be talking of different syndromes, I will not describe which DNA repair pathways it is, because it's a separate lecture completely. But each of these have a distinct DNA repair pathway which is altered. As you know, there are at least 5 which are well described, well researched, and there are other DNA repair pathways as well. So the phenotype is cancer, the phenotype is the appearance of the face, and the genotype is this. But environment we are forgetting. If suppose this young man who was an albino and this man who has xeroderma pigmentosa, right from childhood, they had never been exposed to sun or very little exposure to sun, their phenotype would have been completely different. Except this man, irrespective of exposure to sun, he would have been, his skin would have been like that, but he wouldn't have developed the cancer. So this is the genotype and phenotype correlation. So I'm showing this picture. All of you know this is a famous statue of David, which is in Florence, in the museum in Florence. To explain phenotypes, we when do we look at environment, when we look at the genes, and when we look at the gene environment interaction. So this is, I don't know from which site I got from the internet some years ago. They have shown that if David was growing up and was a young man in 21st century, this is how he would have looked. Okay, hairstyle, I don't know what hairstyle people use it, whatever is of contemporary. So to look at, he is, of course, in those days, the Greeks and Romans, they sort of imagined the perfect body, like the Greek gods and the athletes. So he has been given an athletic body. But here, if he was sort of growing up on the junk food and the lifestyle of the modern men, many modern men, this is how he would have been. So if you want to look at his obesity and we are focusing on his genes, it is completely waste of our time. Okay? So you have to look at his lifestyle, his physical activity, his food intake, and several things. And of course, if there are some people with the same level of physical activity and food, they are not sort of obese, then you have to look at maybe they have genetically they are protected. So then you could have a case control study, small problem. But what has not changed? The shape of his nose would not change. Okay, shape of his nose is, so he has a Roman nose, which is based on his genetics, nothing to do with environment, unless until he was a boxer and getting punched, and then the shape of the nose would change. So when you look at the phenotype, I think that's very important when people do research. Just because in the lab you work on genes, you just sort of focusing on the gene, and you are not sort of collecting data on the other environmental interactions, which could have given rise to the phenotype. If you're only working on a petri dish or this thing, then the phenotype could be, the environment could be what is there in that solution, which is there in the temperature and the oxygen, all those things. So I'm showing this slide. I mean, all of you know DNA, genes, proteins, and proteins have biological function, which talking about, let's talk normal growth and physiology, and cancer is whenever this is altered. Whenever either spontaneously or due to a carcinogen, you may have an altered DNA sequence. I'm using sequence or this thing. What is important to see is that only wherever I've written some. So gene mutation alters structure or shape of the coded protein. Protein alterations change the biological function. Change in biological function results in a disease, congenital anomaly, or evolution. But everywhere I've written some. That means not all gene mutation will change. Gene variations will change the protein shape or function. All protein alterations will not change the biological function, and all altered biological function will not result in disease. There are so many redundant pathways we know. When we work, just talk of DNA repair pathway. There's so many redundant. There's so many ways. There's so much a cell could actually tolerate. You would think that the cell will die. Actually, maybe the cell would die, but an intact human body. That's why we also do in vivo experiments. It is not just in vitro experiment, because in vivo the effect may be completely sort of dampened and you may not see the pathology there, like what you see in vivo. And why I'm saying that, because of this, we see many variants of uncertain significance. What the machines produce when you do genomic sequencing, large scale genomics, they will just tell you this gene is altered, or you call it mutation. Then the bioinformatic pipelines will say this is predicted to alter the to change the structure or shape of the protein and affect this biological function. That's what you do. But finally, what will it do in a human body or in a naturally, not a laboratory bred inbred mice, but in a wild type this thing? So it could be very different, and that's why, and that becomes a big problem for doctors to handle, for the people to report, and for doctors to handle when we have patients. So we have a phenotype and we have a genotype which is not very precise. It is not telling exactly, does this genotype explains this phenotype. Then we have variable penetrance. So we're talking of genes like P53, BRCA, retinoblastoma. That means if they have a mutation, there's a very high lifetime risk of developing cancer. I'm only talking about hereditary cancers, but variable penetrance could be there in most of the Mendelian diseases. No Mendelian disease has, there are few, should not say no, but the penetrance could vary. So these are high penetrance, but then there are low penetrance genes or alleles, mostly from SNPs. Like if you look at, say, GST-P1, null genotype, or CYP1 or SALT1A1. So those who have that particular genotype, that means that enzyme is not working, it is not detoxifying. You might find that those who have their lifetime risk of particular cancer with exposure maybe, say, 15%, and those in whom that enzyme is working because there's no genetic alteration, they also have, but they may have the risk as 10%. So you may say the risk is increased from 10% to 15% to 1.5 times higher risk. So they are giving you some susceptibility, but it's only a slightly increased susceptibility, and that is common for whether it is diabetes or hypertension, and these are generally multiple alleles which will come together. That's why they're called as chronic complex diseases, and gene environment interaction, because they take time to develop and they're complex. So cancer, we know it's a dysregulation of the most fundamental biological process. So anything which is very vital for cell, and that is this thing, except for cell membrane. When cell membrane disrupts, cell dies. Anything else, one of the outcome could be cancer. And factors which govern cell replication and integrity of cellular phenotype can either suppress or accelerate development or progression of cancers. And there are hallmarks of cancer. I'm not getting into that. By Weinberg and Hanahan, they first proposed the hallmarks in year 2000 and 2011 or 13. They came with additional 2 hallmarks. So all cancers that way are a genetic disease, since genetic alteration, that means abnormality in the genes or chromosomes, are a hallmark of a cancer cell. You can never have a cancer cell where the defect is only in the mitochondria or the cell membrane or Golgi or somewhere else, and all the genes and chromosomes are absolutely intact, pristine. That no one has seen such a cell like that. So in that way, cancer is a genetic disease. And the term genetic here refers to somatic genetic disorder. When we think of genetic, we normally think of hereditary, but so it is a somatic genetic disorder. What mutations are there in the cancer cell will be passed on to the daughter cancer cells, and not the hereditary, which is a germline genetic disorder, which accounts for only about 5% of cancer cases where the defect is a germline genetic disorder. So malignant transformation, we know it is a multi-step process, and with each genetic alteration, there's a cumulative effect giving the malignant phenotype. Transformed cells undergo clonal expansion, and sometimes they acquire metastatic potentials. And then we have 2 sort of genes, oncogenes, which are proto-oncogenes, which are activated due to a mutation in a specific domain. And they are like, just imagine, I will show an example, the accelerator of a car which is stuck in full throttle, and it is driving the cell replication in a crazy speed. Anything like any car which is going in a crazy speed with the accelerator stuck has a high chance of meeting an accident, which is like cancer. Or the tumor suppressor genes are like natural brakes. Of course, they have many other functions. They're not there just to suppress tumors. But in terms of cancer, they are like natural brakes of the car, which fail when both alleles are made dysfunctional due to inactivating mutations. Like in the old movies, they used to show that someone will come and cut the brakes, or have a brake fail, and from going from Mumbai to Pune, there will be an accident in the Ghats. So in the multi-step carcinogenesis, again, I showed there are multiple genes. This is a very famous model given by Bert Vogelstein, cancer. So from normal colonic epithelium to adenoma, which is polyps, then carcinoma and metastasis. Basically, you accumulate mutations, and that is how you develop this thing. So this is to summarize, all cancer cells have genetic alterations. It is either somatic mutation alone, which is in the tumor cells, or it is a somatic mutation along with a germline genetic variant. And the variant could be just a polymorphism, it could be risk modifying variant, or it could be a pathogenic variant which has actually caused that cancer to develop. Now, every normal cell in a cancer patient, every normal cell, it will either have only gene polymorphism, because each one of us would have hundreds or thousands of polymorphisms, or risk modifiers only, or sometimes the normal cell would have polymorphism along with the pathogenic variant, which is a mutation. This have a germline mutation. So germline, because it is present in the normal cell, that means it's a germline mutation. Anything which is in the normal cell would also be in the tumor cell, unless until by some mechanism it has been lost. So I'm showing an example of retinoblastoma. For those who are not doctors, might have heard of the RB gene, but what it is, oncologists know that retinoblastoma is a childhood eye malignancy, which is either familial or non-familial. Doctors have known this for many years. Some of them would give a family history, others would not. And what was a non-familial we call sporadic. That means it is not happening in clusters in a family. This is sporadic. So sporadic RB, about 2/3 of the cases are sporadic, and most sporadic, almost all sporadic retinoblastomas are unilateral. That means affect only one eye. As you can see in this boy, so it causes white eye reflex. So it's a tumor which is growing in the retina behind the lens. So you see this white eye reflex, leukoporia. In this child, you see both the eyes have the cancer. So in the hereditary retinoblastoma, which accounts for about 1/3 of all the cases, and majority of hereditary retinoblastomas are bilateral, means affect both the eyes. So that is one thing is known. So this is well-known clinical observations. Doctors make clinical observation, they record, we talk about it in our conferences, and that's where it ends. And of course, it has implication on management. If a child comes to us whose brother also had or father also had retinoblastoma, we know this child's other eye also has at a high risk of developing that. So we keep that child on a special surveillance for a longer period of time. So then I'm showing this how scientists how they can contribute in understanding. And so Alfred Knudsen and his two-hit hypothesis. This is a story. In 1971, he published this two-hit hypothesis of cancer causation, and this explained basically relation between hereditary and sporadic cancer. So it's a very profound. I don't know why it has not got so much of attention. The relation between hereditary and sporadic cancers. He proposed a mechanism of penetrance in hereditary cancers, predicted the existence of tumor suppressor genes and how they should work. And that time they were not known in 1971. And this is recognized as a major contribution to our understanding of all forms of cancer. And actually, Dr. Knudsen, he wanted to be a mathematician, but finally he became a doctor, then a pediatrician, then a scientist. So there are many clinician scientists there. And finally, now he went into public health. So, but I think his little bit of, interest in mathematics helped him to deduce, and I'm showing this picture. So those who into radiobiology would know this curve, and this is so first he plotted the ages. These are the age in month and percent of cases not yet diagnosed or at age indicated. This is retinoblastoma. This line, the dots, the fit on this blue one is those who had bilateral, and he assumed all bilaterals are hereditary, and these are unilateral ones, and almost all unilaterals, and they have, because they have gone up to certain age, that they were non-hereditary. So with the alpha, we know linear quadratic. So this is a log linear plot. If he had put it as a linear and was not log linear, he would not have seen this. So this is straight, whereas this is curved. This has a shoulder and then it becomes straight. And this, it will happen only if there are two events which are happening. So two hit hypothesis, like when we talk about DNA damage. So then that's where we explain many of the effect of ionizing radiation on the normal cell, the shape of the curve, cell survival curve, we explain this. So whether it is the type of ionizing radiation and the type of cells and the radio sensitivity, single hit or double hit, that's what we explain. So he saw exactly the same when he plotted these children who had retinoblastoma, unilateral versus so with this, what he proposed was so his two-hit hypothesis explained. So I will just show what it happens. So he said that sporadic one because both the hits are happening after, two hits are happening, and both because they don't have a family history, no one else in the family gets, both the hits have to happen after birth. And the hereditary one, because two hits, they are born with first hit and they only require the second hit. So here, because they require only one event to happen, second hit once they are born, so they will develop retinoblastoma after acquiring the somatic mutation in the second allele of the RB. And here, because both the alleles are normal at birth, the same retinal cell would have to first acquire the first hit and then acquire the second hit, and that's why. So sporadic retinoblastoma is so rare in the world. If you take, say, how many billion eyes are there, from the age of 1 month to 5 years, and so few, only about say 10,000 retinoblastomas happen sporadic. Whereas the hereditary one, if there are 100 eyes who have a germline mutation in the RB gene, almost all, if not all, 90 of these eyes would develop a retinoblastoma, because every cell in that eye, every cell in that eye, retinal cell, has that first hit. So what it explains is that compared to the sporadic or non-familial retinoblastoma, hereditary retinoblastoma often has a family history because it is coming from the germline, occurs at a younger age, and it is more commonly bilateral and multifocal because the same hit, because the germline, it is there in all the cells of every cell of his body. So this actually, if sometimes for medical students I tell them, if you remember this, any hereditary cancer would have that. They would tend to occur, whether it is hereditary breast cancer, hereditary ovarian cancer, hereditary colorectal cancer, hereditary sarcomas, hereditary endometrial cancer. They will tend to occur at an early age. If it is a paired organ, it will be bilateral. If it is not a paired organ, they might first, after endometrial cancer, then they may develop colorectal cancer, and they would invariably have a family history. So this is because of the two-hit hypothesis. This works for the tumor suppressor genes. For the oncogenes, only one hit is required, and this thing. So this is a difference between proto-oncogenes and tumor suppressor genes for the students. For proto-oncogenes, only one allele is activated, so it is a gain of function mutation, and we also call it activating or gain of function mutation. It's a dominant effect, whereas in tumor suppressor gene, both alleles have to be inactivated. The effect is reduced of this, and it's a recessive at cellular level. When it is inherited, then the mutation, so it is the inheritance is like autosomal dominant Mendelian pattern, but at cellular level, the effect comes only when both the alleles are lost. And why this sort of paradox? Because once you are born with the first hit in one eye or in all the eye, very high probability of that another cell would acquire a second hit by so many causes. It need not be happening exactly the same time. It could be by LOH. It could be the loss of chromosome, whatever, chromosome deletion. So this I was just explaining. So accelerator stuck in full throttle, just one accelerator is there in the car. It's an oncogene, it goes mad. Whereas tumor suppressor gene, the foot brake has to fail and the hand brake both have to fail. And once that happens, then you are in an auto mode and its car is out of control. That's what happens when the cell is out of control, autonomous, keeps on multiplying, then sweat cancer happens. So now the question is it of clinical relevance or only for academics or research? Of course, through research we understand, like what Dr. Knudsen said, someone asked, okay, how does it help? So 10 to 20% of cancer patients will have a genetic alteration either in the germline, which is inherited. So in that case, the family could be counseled about it and along with that person and their future second risk, or in the tumor tissue, which is just somatic. And this could explain why they developed a particular cancer at a young age, their future cancer risk and surveillance for second primaries, if their close relatives are at inherited cancer risk, and if it is a somatic mutation, also some germline mutations like in BRCA and PARP inhibitors, if their tumor would respond well to a particular type of cytotoxic targeted or immune therapy. Now, I'm just very quickly going through it. I've used a chapter for one of my books. So this is very well known example of BCR able fusion gene. So this is a gain of function mutation, and gain of function mutation can be targeted. This is targetable. So imatinib, basically a tyrosine kinase inhibitor. So when it sort of inhibits, and so chronic myeloid leukemia, when we were students, medical students, every medicine ward would have one patient of CML patient, and all medical students will go and see the patient because they had huge spleen coming below the umbilicus, and you will not miss a CML spleen. Now, most medical students are deprived of that, and thankfully, and for those patients, after a few years, they will go into a blast crisis, although it was known as chronic myeloid leukemia. They will go into blast crisis invariably after a few years, and they would die within months. And we used to also give radiation in between, but it was only for palliation. But after imatinib, the course of the disease has completely changed because we are targeting the mutation. And later on, when they started using this drug, imatinib, the responses no one had ever imagined. You started seeing and durable responses, but some patients, after a few years, the CML cells started growing again. So then we realized that the cancer cell had acquired a mutation. And this, although it's a somatic mutation, because this one mutation was causing, so it was like, the only mutation which was causing this cancer. So the mutation was acquired, then new classes of tyrosine kinase inhibitors were, which took into account drug resistance. So you have second line and third line therapy for this thing. So this is about the therapy, understanding the genetics. So we knew Philadelphia chromosome, so this translocation of 922 for many years, but it takes decades of years. So people who work in the lab research, sometimes people ask them, sometimes very rudely, so how does it help? When you say, so nothing, all these things, I think the example is the imatinib example, and there's a Herceptin, which is against the HER2 gene. It took decades from the original discovery to finding a drug which has saved hundreds and thousands, if not millions of lives. So this is like the picture I'm just showing, cancer genetics. When we find mutations, so in genetics it is mostly the heterozygous mutation, and in these I'm listed the high penetrance genes. And in high penetrance genes, the lifetime risk of cancer, depending which gene is mutated, it could be a thyroid cancer in red gene, it could be a brain tumor in say P53, it could be tumor of the eye in retinoblastoma, breast cancer in BRCA1 or TP53 or some other gene, colon cancer, colorectal cancer in mismatch repair gene, or uterus cancer also there, sarcoma, both soft tissue and bone because of P53. So they are a lot of hereditary cancers. Every cancer has a hereditary component to it, and in some syndromes we see a large variety of hereditary cancer. If I take example of germline mutation in P53, and if it is a pathogenic mutation, and if it affects the core functionality of the P53, it has so many functions, then the person can develop, almost 95% of them will develop cancer either in the breast or brain or leukemia or sarcoma or lung or somewhere else. And if they survive the first cancer, they may go on to have second and third and fourth cancers. So how we deal with it, once we know the risk of cancer is there, at what age, in which organ, we can detect it early by doing screening. And for different cancers, there are different screening. For some cancers like ovarian cancer, unfortunately, screening as of now is ineffective. When you use a screening, we want to detect it in stage 1, in a completely curable stage, then only screening helps. Otherwise, you might screen it, but detect it in third or fourth stage, there's no point. So early detection can save lives in many cancers, not in all cancers, but in many cancers. Early detection also makes treatment simpler. So they may not require multimodality, some of them may not require chemotherapy, some of them may not require disfiguring surgery. Then for prevention, drugs with chemo prevention. For some cancers, there is chemo prevention, not for all cancers. Like tamoxifen can reduce the risk of breast cancer if it is a BRCA2 mutation. Some of the COX-2 inhibitors are not really very well proven, but maybe at some stage you would have clear data that they can reduce the risk of colorectal cancers in those who have germline mutation in the mismatch repair genes. So as of now, for prevention of cancer, if you know that someone is surgery, which is like removal of the ovaries, if someone has a BRCA mutation for ovarian cancer risk, or prophylactic mastectomy with reconstruction. So all of you would have heard of Angelina Jolie and what she did. This is the operation she did. And of course it was done with reconstruction. So what do we do? I mean, it's when I showed that, it just doesn't happen like that. I'm just, told you, but it doesn't happen like this. So before genetic counseling is happen, and before pre-test counseling, so before you ask for a genetic test, look at the sequence, whether the mutation is there or not. You have to inform them that, are you suspecting their cancer to have an inherited component? What are the various test options? Facilitate making them a decision, because it has a lot of repercussion. Identify who else in the family could be harboring that mutation, and of course taking the informed consent. And genetic test is not just one looking for gene, and today I don't have sort of the scope in this, but from single gene test to just looking at one specific common mutation like an Ashkenazi Jewish mutation or some Indian founder mutation, to testing multiple genes, or now it has gone to exome or genome also. In some childhood condition you directly do, or or a child is born with certain congenital anomaly, you directly do a whole genome, or you may do a large clinical exome. Clinical exome means you have 400 genes or 4,000 genes, that kind of thing. And finally, once you have the result, you have to disclose the genetic test results, and they're not always very straightforward as you would imagine. There are a lot of gray areas, because I showed there are VUS, there are genes about which it may be on the panel, but the function of which you do not know, or the gene which is showing the alteration, that is actually not described with the phenotype or the disease that you're talking about. That even in very straightforward, for you as a professional to be very straightforward, we know this is a phenotype, this is a gene we have found, this is to be done. But for the person to take it, just imagine yourself, for you to take it, to understand. I'll take a simple thing. If you have a new car, if it's badly damaged, you go to the mechanic, they will tell you, oh, don't worry, we'll do this, we'll do that, it'll be fine. But your just heart sinks. Okay? And here it is your own body, your own life, your child's life, your wife's life, your mother's life. So even though as a doctor, as a genetic expert, you know what exactly is to be done, but for the person to understand it, to internalize, to accept, and to go along with it is not so easy. So they may require a lot of psychosocial support. And then while doing that, finally, you have to help them come up with a plan, because ultimately, you can't just freeze. You can't just freeze like, you shine a bright light in front of a deer, they just freeze. They can't just freeze. They have to move ahead and they have to do something. But this all would happen that someone suspects inherited predisposition and refer for genetic counseling and testing. Unfortunately, in many countries where the doctors are very busy, they haven't been trained in taking detailed history, because during training also they were trained in a very busy hospital. That does not happen. So because doctors only will suspect, the genetic testing people or the researcher will not directly be coming to the outpatients department or the wards. The doctors will see them. So that doesn't really happen. So in genetic counseling, there are many principles, and I have learned it myself only by reading, but I can vouch for that each of these principles have a fundamental value. Whenever you breach these principles, not in all patients, but in some patients, they actually would be damaged. Damage is not always physical. Sometimes it is physical also, emotional, relationships. There's so many ways a person you could harm. And in Latin, they say for medicine, primum non nocere. That means first do no harm. So genetic counseling and genetic testing is a double-edged sword. It can cut both ways. So it has to be done with empathy. You have to have beneficence, non-maleficence, non-directiveness, help them make an informed decision and maintain confidentiality and privacy. What I did not say, in India, unfortunately, many patients, like suppose a lady, generally the husband will come and decide, or an old person, someone's father is 75, the son will come and decide. Or so the autonomy, everyone does not get the autonomy. Then informed decision, confidentiality. Sometimes they will send the driver to collect the genetic test report, and and privacy. It is not, but there are areas where if you breach confidentiality and privacy, there will be repercussions for that family and for that person. Non-directiveness, our patients are so used to doctors telling them, you do this, you do not do that. If you just explain to them options, sometimes they get confused. Why is it just talking about options? So you have to do it in a culturally sensitive manner. So making syndromic diagnosis and differential diagnosis, basically the types of cancer, age at cancer diagnosis. When we talk of cancer genetics, that unilateral, bilateral, histology features, who all are affected, and there are many other phenotypic features, polyps, skin, eye, cyst. So remember what Knutson's hypothesis. I'm showing this slide because I just want to highlight one thing, that doctors are already facing lawsuits that broach a new legal terrain, and this is in US, and everything that is in US will finally come to other countries also. Sometimes they even hold them liable for how they offer, interpret, and counsel patients about genetic tests. So that is happening, and this is a malpractice. This thing. So this lady said she was damaged for the rest of my life because surgeons mistakenly removed her breast and uterus, because nurse told the surgeon she misread her BRCA genetic result as positive. And so I don't know what exactly she read, and she had a prophylactic surgery. When her family member was asked to do that, their doctor told that no, in your family this is not a mutation which will cause. So I remember I said that pathogenic mutation or a VUS, and what actually it means. So I mean, it could be a serious issue. This is an old case. This famous doctor of New York, Dr. Pate, Dr. Pack, he was the leading surgeon in New York those days. He operated upon a patient who had a familial adenomatous polyposis, didn't tell anything to the family. The patient died. He also died. The patient's daughter later on developed colorectal cancer, and she sued his estate. Says that our family should have been cautioned about it, and I would have been screened, and I wouldn't have been diagnosed with advanced cancer. So coming to India, I'm talking about just India, the South Asia as a subcontinent, because we have one large gene pool. We have 1.5 billion South Asians. We represent a quarter of the world's population, and 1.6 million new cases. And even if you take just 3% of these being hereditary, we have about 50,000 familial cancers per year in India. So with 4 members per family, you would have about 200,000 people who require all that I said: genetic counseling, genetic testing, surveillance. And just those of you who have visited Tata Hospital, you will require few Tata Memorial Hospitals only to cater to this population. Unfortunately, there's only one single comprehensive cancer genetics unit for this entire South Asia, which is ours, which we provide everything. There are other centers who have started. I'm very happy in the last couple of years, but they have not sort of yet become comprehensive in terms of having a daily cancer genetics counseling clinic, risk assessment, genetics lab with which they can talk, go back and forth, counseling for all kinds of things, and risk management. And even our is also not really that much. So oncologists basically have to get into this. So we have now over 9,000 families, and as you see, the most common syndrome is hereditary breast ovarian cancer. This is the name of the syndrome, and then bracket the gene which is mutated. So BRCA1/2 causes HBOC, mismatch repair, hereditary non-polyposis cancer syndrome, or Lynch syndrome is the correct name actually now. APC, P53, Li-Fraumeni syndromes, multiple endocrine neoplasia, red gene, neurofibromatosis, albinism, XP. So there are many. So if you look at most of the genes which are listed in the site, they are involved in DNA repair, different pathways of DNA repair. So we have to do pedigree, phenotyping, counseling, molecular genetics, and finally risk management. Now in Tata Hospital, we see every day close to 5 new families, and 10 or 15 of them come for follow-up. Last year in 2019, we had more than 1,500 new families, and this year, of course, because of lockdown, we'll have fewer patients. And so this shows the need for cancer genetics. Unfortunately, majority of the hospitals, even cancer hospitals in the country, do not have cancer genetics clinic, and I don't blame them. They will not find cancer genetic counselors, and the oncologists, practicing oncologists, are so busy, and they haven't actually had the training also. So these are the individuals we had in our this thing who were tested, and we have found mutations in over 1,600 individuals and 1,000 families, and in our lab we test about 18 genes, and there are many. So the ones in the green are the ones who were tested negative for the mutation which their mother or the sister or the or the brother or the father had. So they have been really benefited from that. There are lots of ethical, legal, and social implication. I'm not getting into detail, but all I would say is the myth is that we have one large family where everything is shared. It is just a myth. You have to treat each family and each person as an individual. They may have some common issues, they have some unique issues, and I think that you have to deal with it. So through our work, we have found that many of the mutations, specific mutations, they are common from a particular community or a region in South Asia. And what we call, we have done haplotyping analysis, my colleague Pradnya, and through that we know these are founder mutation. That means they had a common ancestor who actually had the mutation, and over the period of generation, because people married within the community or sub-community, there are many people today who would have the same mutation. Because I'm just showing the penetrance, the lifetime risk of cancer. Like this is the most common mutation that is in our cohort is 185 del AG in BRCA1 gene in breast cancer. It is also known as the Ashkenazi Jewish mutation, but in India it has arisen independently. Lifetime risk of breast cancer is 75 to 80%. Lifetime risk of ovarian cancer is close to 40%. So these are serious things. People used to say, but because we have curcumin in our diet, because there are more people are vegetarian, women marry early, so they will not get cancer even though they have mutation. Unfortunately, when a mutation is in a gene like high penetrance gene in a BRCA1 or BRCA2, lifestyle is offering very little protection. So this is more for the doctors. I believe there are not too many, medical doctors. So when we have a newly diagnosed breast ovarian cancer with suspected hereditary breast ovarian or BRCA mutation, we have a higher index of suspicion for multicentric or bilateral disease. So we may do MRI in these patients if they are young, or on mammography the breast is dense, we can't see it. And therapeutic approach in terms of treatment, we discussed that the indication for genetic counseling and testing, and that tell the results will come after therapy. Even the private labs, if you send it today, the report will come in about a month's time, and no cancer patient we keep on hold for a month to start their treatment. And the other things which are more for focused, breast oncologists, which I don't think in this group are many. So about breast conservation versus doing a mastectomy, and then doing prophylactic mastectomy. And one thing in terms of the therapy is that patients who have a germline BRCA mutation, because they have a defect in the homologous recombination deficiency, this thing, what is a term called as synthetic lethality. So if you inhibit the single strand DNA repair pathway also by drugs known as PARP inhibitors, then these cells die very easily. So those who have a germline mutation in the BRCA1 or other genes involved in the HR pathway, they respond very well to PARP inhibitors and also platinum drugs. And during the follow-up, you have to, of course, counsel them about, if they haven't been counseled, you send them for detailed genetic counseling if you're lucky in your center app, or you can send to us. We also now, we found doing counseling on Zoom or through Google Meet is quite sort of useful. They need lifelong surveillance. Before that, I say not just the patient, because you're dealing with the patient, but she has a sister at home. She may have a daughter who is now 19, but in few years' time she'll be 23 or 25. She may have a cousin who may be at risk. There's so many people, or a brother or a child. So they all also have to have genetic counseling, risk estimation, and then genetic testing, and then surveillance. And they require lifelong surveillance. So like in a breast cancer, for the contralateral, which is the other side, and ipsilateral breast cancer risk, and mammogram is not sufficient in most of them if they are young, so MRI. And if risk reducing salpingo-oophorectomy, which is an operation to remove the ovaries and the fallopian tube, then you explain to them that screening with CA125, which is a tumor marker, or ultrasound is really a useless thing. It is just for keeping them false reassurance. Chemo prevention or risk reducing surgery as part of study, and whenever if they have a recurrence, then consider this thing. And healthy women, I'll just tell you, whatever few oncologists, that as oncologists, we do not know how to deal with healthy women because we've never met with healthy people. We only deal with cancer patients. So any kind of cancer, any stage, anywhere, we will be able to maybe a little bit of just refresh. If it is an uncommon cancer, we will know how to deal with it. May not be technically always, but in general, how to approach that patient. But healthy people, they ask differently. Their thought process is different, and the concern that they have is very different. So they would require and this, I think, they should be dealt with by genetic counselor or high risk clinics, if you are lucky to have them, or we could help them. So they have to have a detailed genetic counseling. So about breast cancer surveillance, monthly breast self-examination, 6 monthly examination by the doctor, and annual breast imaging, sometimes alternate mammography with MRI. Discuss what I've said, limitation of ovarian cancer screening. Everyone wants to get screened, no one wants surgery. And then risk reducing surgery. And other thing too important to say that I had some patient who were tested negative and later on they developed breast cancer. They were very upset, doctor. They thought they have got the wrong report. That even when family specific mutation is not found, they still are at population risk of that cancer. And awareness and screening for breast cancer is appropriate. So that is important to say. And I'm just there are several scenarios which we use for one of the storytelling. Whenever there could be consequences on individual and their family when principles of genetic counseling are ignored. I won't go into the details, we don't have time for that. Then colorectal cancer, I'll just very briefly talk about it. It could be hereditary, familial. Familial means it's not like a classical Mendelian or sporadic. And it is either polyposis with lot of polyps in the bowel or non-polyposis. Polyps could be adenomatous polyps, which the pathologists tell us, and they are either familial adenomatous polyps, which I showed you the legal case, or autosomal recessive MYHS, or hematomatous polyp, which is jumbled up, tissues, normal tissue, Peutz-Jeghers and Cowden and other juvenile polyposis. Or it is a non-polyposis. It may have few polyps. It is autosomal dominant, which is Lynch syndrome, quite common, not very common, fortunately, or autosomal recessive, which is very rare, called constitutive mismatch repair deficiency, when both alleles are mutated. In all these, only single allele is mutated. So this is how the FAP, multiple, you can see this round thing. These are polyps, and these are the two cancerous growths over there, which is there in the resected bowel, which has been opened up. And they could also have its own phenotype, like those who have a FAP, they could have thyroid cancer, brain tumor, medulloblastoma, liver tumor, benign tumors of the jaw, osteomas, desmoid tumor of this thing, and this is a retinal pigment epithelium. So they could have various things. So doctors have to sometimes making a syndromic diagnosis, even if the APC gene is not tested. But if you have more than one feature, or someone has hundreds of polyps and jaw osteoma or a desmoid tumor, the only diagnosis is FAP. And surgery for FAP is quite extensive, and I'm just showing this. I'm not going to describe this, just showing that even to prevent cancer, because there are hundreds and thousands of polyps, the entire rectum and colon is sort of full with that. You can't just be doing endoscopy every month and resecting them. So before they develop cancer, they remove the entire colon and rectum. They try to prevent putting a colostomy bag, but sometimes even that is required, and someone who doesn't have even a cancer. So a lot of issues are there. They require special counseling. So we had our cohort, so we found many, and of course in India, so little work has been done. Any syndrome that you work on, you will find novel mutations and phenotypic association. So it is not surprising. I won't go into the details. Lynch syndrome, it has few polyps, and I'll just talk about and the extra colonic cancers. Again, people don't appreciate that they have endometrial cancer risk. Sometimes even gynecologists in our country do not always appreciate that if a lady has endometrial cancer and gives a family history, say her father or her brother or her sister had colon cancer, what you're looking at is a classical Lynch syndrome, especially if it is early onset endometrial cancer. And other cancers could also happen in 5 to 10% cases. So microsatellite instability, I will not. So I'm just showing in use of things which could be done easily and quite well. Before that, what I was showing is looking at microsatellite instability. So comparing the DNA from the lymphocyte, which is the genomic DNA germline, the DNA from the tumor tissue, and you will see, because you're looking at fragment length analysis, and what you find that because of the mutation in the mismatch repair gene, any repetitive sequence is more, they're likely to have a slippage, and you would have a unstable. So microsatellites become unstable, microsatellite high, MSI high. But on immunohistochemistry also, you can infer that those who have a mutation in, say, MLH1 gene, in those patients, the MLH1 protein will not be properly formed, and it will not be expressed in the normal cells of the colon. So we found that the IC-based MMR gene testing was very, this thing, we had about 159 cases, and majority of the patients who had a loss of a particular protein, say MSH6 or MLH1 or MSH2, most of them had, we found a germline mutation. Of course, this was done in a validated, in a accredited lab by expert GI pathologists. So if you are not getting, maybe it is not being done that well. In Lynch syndrome, all lifetime, like I showed for breast cancer, they are close to 80%. Any cancer, MLH1 and MSH2 gene, the most common gene. For colorectal cancer, risk majority is there. And you say the colorectal cancer risk starts from 25, 30 years of age. Endometrial cancer risk is close to 50%. And of course, this is another syndrome called Peutz-Jeghers syndrome, which we described last year. So again, the phenotype description is very important. If you're not describing it, then the doctors will not be able to pick. I won't go into the details of it. NGS, all of us are using NGS quite a lot. It makes life easier. It can pick allele dropouts, which can be missed on NGS, but it will also identify more patients with VUS. So in our own data, because very few Indians have undergone, whole genome or whole exome or large panel gene testing. When I say very few, only tens of thousands, not millions. So what we find that in our patient population, about till 2 years ago, about 1/4, irrespective of how strong the family history is there would be a VUS. These days, we are seeing around, say, 10 to 15%. And I think when more data comes, Indian data, genome variation data comes, then maybe it will come down to 5%, which is there for the Caucasians. Now, this is something I wrote few years ago. Why I'm bringing this up? Ethics and clinical utility of direct-to-consumer genetic testing. And a lot of you work in research lab, and you have a sort of curiosity. I'm working on P53. Okay, someone will donate their blood. That happens, and you will just look for P53. What if you find a VUS? What if you find a pathogenic mutation? So pathogenic mutation, of course, person can benefit, but if you find a VUS and you say there's something in there, you go on the database search in ClinVar and another thing, you will say this is variant of uncertain significance. So it can, and when you do a direct to consumer testing, so there was 23andMe, and now they can only report about ancestry and share the raw data. So it can have a lot of ethical aspects, and it can create havoc. I think technology is there. Any one of you want, in few $100s, you can get your entire genome, germline genome tested at a low sort of depth, say 10x or 20x. But does it help you? I think it may harm more of you than help you. So with this, I'm sort of ending. So we have a genetic clinic which runs in Tata Hospital, 3rd floor, 305. And we have, it is a snake and ladder of genetics. So our lab, Pradnya, who's with me, she has done a lot of genetic testing. ICMR, DBT, Ravi is our cancer genetic counselor, and many others before him have been there. We have 9,000 families. I mentioned only 2 students. This I had made for a colorectal cancer. So Nikat and Anuja, but before that, Vasudha, Mukit, and several other people, almost more than a dozen people who have worked in the lab for many years who have produced this data. Thank you.
Thank you so much, sir. And so Dr. Vijay Singh will take it from here.
Thank you, Dr. Sarin, for wonderful talk. It is already 5:20 at your end, so I would just ask to stay 10 more minutes to ask few questions.
Okay.
And unlike online, there won't be follow-up question from the person, so you will keep that in mind. So first question is from Dr. Kinja Sarkar. Does derma pigmentation cause brain damage also?
Yes. So there are several types of XP, XPA, B, C, D, E, F, and some of the XP, because they are different genes. So some of the types of XP could be associated with mental retardation and other phenotypes also. Not all of them, but yes, it could be.
Next question is from Shahana Ghosh. How mutation in BRCA2 gene leads to different cancers like breast cancer, ovarian cancer, and pancreatic cancer?
Okay. So when you compare BRCA1 and BRCA2, BRCA1 because that was the first gene which was found was called BRCA1. The second one which was found a year later was called BRCA2, which causes hereditary breast ovarian cancer. But we know that BRCA1 and BRCA2 are slightly different. And of course, in both the genes, the main cancer risk is for breast cancer and ovarian cancer. But in terms of the phenotype, the breast cancer in BRCA1 is invariably triple negative, whereas in BRCA2 gene, the breast cancer, many a times it is a ER positive tumor, it is not a triple negative. That's why chemo prevention with tamoxifen or oophorectomy works better for BRCA2 gene. Plus, in males, the risk of cancer for male breast cancer and prostate is higher in BRCA2 mutation carriers as opposed to BRCA1. And the other cancers like pancreatic cancers and other cancers are also more in BRCA2 as compared to BRCA1.
Next question is from Arun Hose. If all gene mutations are not leading to cancer, does it mean that only focus on certain potential oncogenes when we talk about cancer development?
See, your question is right. We know that not all mutations, but our knowledge about the number of genes, mutations, and which can cause cancer is increasing. So, like, let us give an example of hereditary. I mean, we knew for hundreds of years that breast cancer could run in families, nothing new. Then, once the first gene was found, which was BRCA1, that explained only about 50% of, or less than 50% of the families which had that hereditary cancers. Then BRCA2 explained another 20-30%. Then P53 explained anything. And now after that, there are 8 or 10 genes. Still, there are about 10 to 20% of families where we know that they are hereditary. It is running in the family. It is Mendelian. But when you test for these genes, you do not find any mutation. So, of course, once all the low-hanging fruits are gone, the more you search, it becomes a law of diminishing returns. Plus, because you're looking at the genes of an individual, you might find many variants about which you do not know. So the psychological impact on that person, the psychological harm, that you have to take care in mind. If that was not the concern, I would just for everyone look at the whole genome or whole exome. But you have to finally tell the person, and how that person will take it. You don't want to confuse them more than it is necessary. That is my answer.
Next question is from Professor KP Misra. Are there identifiable set of genes which are more sensitive to mutation by radiation or chemicals?
Okay. So, I mean, it is the region. Some DNA sequences are stretches of DNA sequences are more sort of prone to mutation, like repetitive sequences. So, majority of the mutation, if you look at the spontaneous mutation, we do not know how the germline. If let us say, I'm giving example of BRCA1 only. Let us say because it is most well-known gene. A lot of these mutations are insertions or deletions or truncating mutations, and they are in repetitive, like you have AG. There's a repetitive 3 times it is repeated, 1 AG is gone. Or a mononucleotide repeat, say AAA 6 times, 1 A would disappear. So, I mean, it is like, it is known as in the tumor cells or the genes which are particular mutated because of ionizing radiation. It is not like that, but those who have a particular type of genetic defect, like a mismatch repair or ATM gene is mutated, so they are more likely to have a more genomic instability, and especially after DNA damaging agent like ionizing radiation or certain type of drugs. So patients who have XP or something, some of the drugs that you use for them, or ATM or ionizing radiation, you have to be a little careful because they may have more profound effect, especially on normal tissues.
Next question is from Pradeep Misra. How do you see epigenetic gene environment interaction as treatment of the breast cancer?
So today I was talking of only the genetic, not the epigenetic, but epigenetic cannot be ruled out because in the beginning I mentioned gene regulation. So we know the epigenetic changes, the post-translational modification, there are so many things which happen. And now epigenetic alterations are being targeted. So first of all, it's a modifiable life, risk. So, there are certain things if you avoid or do that, you are less likely to have that epigenetic, effect. Plus, even the epigenetic changes, there are many things which they are being targeted. HDAC inhibitors and other things are there. But what in gene environment interaction, definitely epigenetics has a role. I mean, it cannot be denied. But the extent to which it occurs, how much of it is reversible, what are the condition, and because the reason is in studying complex chronic diseases in vivo is so difficult. And that is the reason why we haven't been able to dissect out completely.
Last question is from Tejinder Kataria. What is the significance of uncertain variances after genetic test?
So uncertain significance, I think in the beginning, I think one of the main reasons for doing genetic counseling before the test is to explain that one of the outcome of the genetic test could be that you find a result which is neither says all clear or there's a mutation. All you say is we have found something about which as of now no one can say whether to ignore it or to take it into account. In some families, we can actually look into it, and we can say that. And it so happens, as of now, majority of the VUS later on turns out to be harmless. That means nothing to be done. But about 5 or 10% of these VUS, in a few years' time, we know that they are actually the cause of this cancer, and they should have been acted upon and all that. So in VUS also, you can classify them different categories like VUSD and other this thing. So VUS requires special counseling. Patient, especially the people who have low hereditary cancer risk, and they insist for genetic testing. And if you get a VUS, and especially that person has a type A personality, the counseling is going to be very difficult. So you prepare them in advance about the possibility of a VUS, and depending on, first of all, the VUS is in the unrelated gene for the phenotype. It is easier to discard that. But if it is in the same gene which sort of there, then you have to take it more seriously.
Now I would like to thank you for your outstanding talk. We have received truckload of appreciation from viewers or audience under comment section. Thank you very much.
Thank you. Okay.
So I would now like to invite Dr. B.N. Pandey, Secretary SRR and Head Radiation Signaling and Cancer Biology Section, RBNHSD, BRC Mumbai, to present the memento to Dr. Rajiv Sarin.
Virtual
okay. Memento, yes.
Yeah, I mean, not virtual, it is real. So,
okay.
On behalf of society, I like to present this memento virtually, but it will go physically to you. We just within few. Yeah. So this will be. Thank you for joining. Yeah.
Thank you.
And the memento to Dr. Singh, Vijay Singh, sharing this session. So this also will be sent to him, or maybe whenever he will give it. So
no, I will come next time and collect it from you.
That will be better. So this is the memento. So ethically it is all ready. Only we since due to virtual we cannot give. So before I, Pooja, can I just tell briefly about maybe 2 minutes about the school we are going to organize? It's a School of Radiation Research.
Sure, sir. I will share this. I will share the slide.
Yeah, just you want to share or I should share it?
I'll share it, sir.
Okay. So basically, this Indian the Society for Radiation Research we organize alternate year the International School of Radiation Research. So last school was organized in 2017 in Annamalai University, first International School of Radiation Research. But now this year, due to COVID, we cannot do this school 2020. So we are going to have a online e-conference mode. This International School of Radiation Research. So that will be our second International School of Radiation Research. Pooja, can you show this slide?
Sir, I am not able to share it. You will have to.
Okay, let me do it. Don't worry.
Yeah.
Just a minute. Yeah, I'll do it. Just I got only one slide. So, can you see whether my slide is visible?
No.
Yeah. No, I think it is coming now. Sorry. So,
Sir, it's not visible.
is visible.
Yeah, now it's visible. You can make it full screen.
Maybe this slide is visible.
Yeah, you can make it full screen.
So this its mechanism and human health implications. So this will be for 2 weeks basically. And this will be every day 2 hours. Yeah, slide is visible, Pooja?
Yes, sir.
Should I make a full screen? Okay.
Yeah,
Full
make it in the
screen.
presentation mode. Presentation mode.
Okay, okay. Can you see? Yeah. So this school will be for September 6th to 20th for 15 days, and time will be evening Indian time, and it will be through e-conference mode. And we are going to have this time. It is going to be very limited participants, maximum 95. So those audience who are listening and if they want to attend, once we announce this on the SRR web page, they should register within time so that they don't miss the chance. And we are going to cover major topics related to the theme of the conference. And we are already inviting very eminent scientists from India and abroad, which are related to this theme. So we are going to have not only the talks, but also we are going to have few sessions on different techniques about DNA damage quantification. So, and we got confirmation of few foreign speakers and who will give the talk, and also some Indians eminent scientists they are going to give. So this is just a small announcement. So to join this school, and this is going to be very useful for young faculty, students, and postdoctoral fellows. Thank you very much.
Thank you, Dr. Pandey. Now we will just end our session. I'd like to invite Dr. Nagraj to give a vote of thanks.
Yeah, thank you, all of you, for joining this webinar. And Dr. Vijay Singh, special thanks to you for chairing the session. And it must be very early in the morning for you, and you took the time from your busy schedule. Dr. Rajiv Sarin. Rajiv is a dear friend and as always an excellent talk, and I also learned quite a bit. Thank you, Dr. Rajiv Sarin. Dr. Sham Srivastava is the president. I mean, he was there at the beginning, and Dr. Mishra has been there. We thank both of you for managing SRR. I also would like to thank Mr. Deepak Naik, who prepared the e-certificates, and Pramod Meher from Germany, who prepared our Facebook site. And of course, I special thanks to Pooja Melwani, who is conducting this meeting, and she has put in a lot of hard work. And of course, I'll be failing if I do not acknowledge Dr. Pandey's contribution both to SRR and for this meeting. Thank you, Rajiv. Thank you, Dr. Singh. Thank you so much, and we meet again for the next meeting. Thank you.
Thank you.
So thank you, everybody. I just wanted to announce one thing, that all the participants can download their e-certificate from the SRR website. So we have added a link to the web page, and you can enter your registered email ID and download your certificates. Thank you for participating. Have a good evening.
Thank you, everyone. Thank you.